Okay, so now we've covered the basic chemical structures of the nucleotide. Now let's discuss how these nucleotides come together to form a polymer. So nucleotides are covalently bonded to each other to form single-stranded DNA, and these use phosphodiester bonds. So nucleotides are linked together by phosphodiester bonds between the phosphate group at either the C5' position and the OH group on the C3' position. So let's just see how this works. So here is an example of two nucleotides that have been bound together. So this is the 5' end. So again, 1, 2, 3, 4, 5. This is the 5' carbon where the phosphate group is attached. And so now we have two of these nucleotides that are bound together. And so the phosphodiester bonds that we refer to are the bonds between the carbon here and the oxygen here, or the carbon here and the oxygen here. So one of the things I encourage you to do is actually look at this and this in more detail in the free sugar form, just to compare what are the atoms that are still present. So the formation of these bonds between the phosphates actually releases molecules, releases atoms, and so being able to distinguish between that is an important part to make sure you understand how this process works. And so here, we link phosphates together, and we can continue on to do it in a linear pattern. So here we have 1, 2, 3, 4, 5 that are grouped together, and the nucleobases are different. So the A, T, G, C, A, these nitrogenous bases are all different, and so this is where the information is encoded. And this also actually determines the order. So when we look at this linear string, how do we know it's A, T, G, C, A and not A, T, C, G, T, A? And so the way we read this, we talked about this a bit before, is that we read things in the 5' to 3' direction. So this is the 5' to 3' direction. We start from the 5' of this sugar, and we go towards the 3' of this sugar, and so this determines the direction of the strand. So you notice all these all these sugars have the same orientation of the 5' and 3', and so in this case we read T and then G. So the structure of DNA was further determined by 3 key experiments, and so this is how we go from having a single-stranded piece of DNA to a double-stranded piece of DNA. And so one of the key experiments was work done by Chargaff between 1949 and 1953. And what he did is he measured the amount of A, C, Gs, and Ts in samples, in biological samples, and found that there was an important relationship. So the amount of A is proportional to T, the amount of C is proportional to G, but the percentage of C and G does not equal the percentage of A plus T. And so this he was able to infer, and so this was a very key set of information that future researchers were able to use to infer the complementary of DNA. Rosalind Franklin in 1953 did very important experiments looking at the structure of DNA, and was able to demonstrate that DNA was a double helix. And then using this information, Watson and Crick were able to propose a structure of DNA. So Chargaff rules. So here, if you look at a bunch of different biological sources, so this is the original Chargaff data from a 1950 paper looking at various organs of ox, and also different organisms as well. And so what he found is that when you compare the A and the T and the C and the G, they're approximately the same. So this is there's experimental error in here. And kind of future work has shown that is much similar. So A and T's, they're all very similar, and then the C's and G's are all very similar. But what you notice if you look in different organisms, there's a huge difference in the amount of A's and T's. So the humans has approximately 60% A's and T's, whereas if you look at Sarcina lutea, I have no idea what that is, but that's only about 25%. So this is a very GC-rich organism, and this is also shown here in this figure, the amount of G's and C's. So X-ray diffraction studies, so studies by Rosalind Franklin, showed that DNA had a 34 angstrom periodicity, And this pattern that you see right here is what you would expect if DNA was actually a double helix. So by diffracting DNA, diffracting X-rays, by running it through specially prepared DNA samples, the diffraction pattern gives you information both on the shape, so this is X is the shape for a double helix, and then the spacing of these also gives you information on how repetitive this is. And so based upon this information, Watson and Crick proposed a model back in 1953. So they proposed that DNA was double helical, which agreed with the Franklin experiments. And then they also proposed that it was two anti-parallel strands that were connected together by base pairing. And so the outside has two different backbones, so we can see a backbone kind of running through this, but you note that there's actually two different backbones going on. And so if we look at the directionality of one would be in one direction. And now if we run through the second strand right here, what we would find is that directionality is the opposite. So this one would be the 3' end, and this would be 5' end. And so this is what we mean when we talk about anti-parallel. So if they're parallel, the 5' and 3' directions would go the same. Anti-parallel means the two strands run in opposite directions. And so the reason that these are connected is not because of covalent bonds. So these are not covalently bonded together. You could actually separate these two strands pretty easily. The reason that they're bound together is by hydrogen bonds, and these are formed between the nitrogenous bases. And so again, remember this backbone is very flexible, but the nitrogenous bases are very rigid. And so they're able to come together, and if the orientation is just right, they can form hydrogen bonds together. It is also a right-handed, the helix is right-hand, so this determines kind of the directionality of it. So, you know, if you follow the direction of it, so you can follow this in two directions. So if you put your thumb down in this direction, so basically if you rotate around, so you can see that, you know, at this point right here, at this point right here, you know, the DNA, the blue strand is kind of wrapping around this way. And so the directionality goes down. So as this goes in that direction, it also goes down. You could also do it from the red strand. So the red strand is wrapping around in the opposite direction. So it's wrapping around like this at that point, and it's going up as it wraps around this way, it goes up. And so that's why we say it's right-handed. So if you had to use your left hand in order to get that right, then we would say that DNA is left-handed. If you want to be very pedagogical, whatever that word is, you know, you'll notice that a lot of pictures of DNA that you see in the news, they actually show DNA in the improper way. So if you see it, you might find a left-handed DNA helix instead of a right-handed DNA helix. And then the base pairing, the chemical affinity produces the hydrogen bond. So this is where the Chargaff rules come together. Because if you have double-stranded DNA, then that means for every single A in one of the strands, you have a complementary T. And then the same thing is true with the GC pairing. So you can draw any sort of sequence of a single strand you can think of. So you're free to do whatever you want. So in this case, we have a strand that has a lot of C's. So 1, 2, 3, 4, 5, 6 C's. This has 2 A's and 1 G. But the complementariness means that if we now draw the opposite strand, this would be a G, C, G, 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 T, T. And so we would have 6 G's here, 2 T's here, and 1 C. And so if we add this up, there's 7 C's, right? This and this. There are 7 G's. So that's why there's always an equal and opposite amount of G's and C's. And that's why there's always an equal and opposite amount of A's and T's. And then you should also, part of the activity will actually be to force you to kind of look at what are the bonds that are formed. So what you need to look for are things that have negative or positive partial charges. And so these are the nucleobases. So the A and the T. This hydrogen right here has a strong positive charge. This oxygen right here has a strong negative charge. This nitrogen has a strong negative charge, and this hydrogen has a strong positive charge. And so what we have right here are two different hydrogen bonds. So A and T form two hydrogen bonds together, and that's what holds the double strands together. And then the C's and the G's, they actually have three different hydrogen bonds. And so again, in each case you have one purine paired with one pyrimidine, one pyrimidine shared with one purine. And so the G and the C connection is much stronger than the A and the T, and that's because there's two bonds versus three bonds. And you can always orient yourself by looking at the C1 prime carbon. All right, so the alternate forms of DNA, the final thing that we're going to cover in this part is that there's actually different, there are different ways that the helix can arise. So there's A, B, C, D, E, P, and Z forms. And so what is meant by this? And so because the backbone is so flexible, there are actually different geometries by which these bases can actually line up. And so this is an example of the B form of DNA. This is the A form of DNA. And so if you actually measured the distance, so if you went one full cycle of the helix, so this is actually only part of the cycle because this strand goes all the way to here, so you'd have to go all the way down to here, and you compared it to this, you would find that the sizes are actually different. And so the thing that determines the form of the DNA is how far down you have to go, how much vertical direction you have to go before you do a full turn of the helix. So the Watson and Crick DNA molecule model was of B DNA, and that is seen under aqueous low salt conditions. The A DNA is prevalent under high salt or dehydration conditions. So the conformation of DNA is largely determined by the chemical environment it finds itself in. So is there a lot of salt, a lot of water, etc. And then also other things that can induce different forms is the nucleotides that are actually within it. So some of these require certain sequences of nucleotides in order to form these different geometries. But the main thing I want you to remember is the fact that DNA can kind of take these different forms, and there's kind of a fixed amount of changes that it can have, and that's because this backbone is flexible, and so it can kind of mold itself around until it finds another shape where these bases can line up together.